Hello, everyone. Welcome to our class on the serologic and molecular detection of bacterial infections. I'm here once again, Miss Merle, and I'm thrilled to have you here. So today, we'll explore two key methodologies, the serologic techniques for detecting antibodies and antigens of bacteria and molecular methods like PCR and next generation sequencing, but we will not be talking much about next gen. And then we'll discuss their principles, clinical applications, and their importance in diagnosing bacterial infections. So feel free to engage and ask questions as we dive into this vital topic. So thank you for joining or listening, and let's get started. So for the topic outline, we will be talking about basically four major bacteria. So we have group A, streptococci, we have the pylori, Mycoplasma, pneumoniae, and Rickettsial infections. So let's start with the immune defenses against bacterial infection. So with our innate immune defenses. And then the physical barriers, of course, we have the skin and the mucosal surfaces. So intact skin and mucosal surfaces, they serve as the first defense or the first line of defense against pathogens. We also have soluble factors in the form of lysozyme in tears and saliva. They break down bacteria and other soluble factors, including interleukins, prostaglandins, leukotrienes, and acute phase proteins like CRP, haptoglobin, and ceruloplasmin. They coat the bacteria or eliminate substances that encourage bacterial growth. So we also have another innate immune defense, phagocytosis. So the neutrophils, monocytes, and macrophages, they ingest, they engulf and destroy bacteria. And the alternative complement pathway enhances phagocytosis by recognizing microbial cell walls. And for the adaptive immune defenses, we have antibody responses and the cell-mediated immunity. So antibodies, guys, they neutralize bacterial exotoxins and they form immune complexes for elimination. So the classical complement pathway is activated, leading to bacterial cell destruction or lysis and increased inflammation. So antibodies also enhance phagocytosis through opsonization. Then let's move forward with cell-mediated immunity. So this is essential for targeting intracellular bacteria like Mycobacterium tuberculosis and Listeria monocytogenes. So we have CD4 and CD8 positive cytotoxic T lymphocytes. Basically, CD4 positive T cells, they are for macrophage activation, and for killing the infected cells, we have CTLs or the cytotoxic T lymphocytes for that. So mechanisms of evasion, how these bacteria can evade the immune system responses. So we have evasion of antibodies. So by antigen variation. So certain bacteria like streptococci, they mutate their antigens, rendering host antibodies ineffective. They can also downregulate, or there's this downregulation of MHC molecules. So the bacteria can decrease MHC molecule expression. This can really hinder T cell recognition and response. And some bacteria, they can degrade IgA, such as Neisseria gonorrhoeae, Haemophilus influenzae, and Streptococcus sanguis. They produce proteases that break down, is the protease that break down or degrade IgA antibodies. So evasion of phagocytosis, inhibition of chemotaxis, interference with adhesion and capsule formation. So for chemotaxis inhibition, Pathogens like Neisseria gonorrhoeae, they block the release of factors that attract phagocytes. While Streptococcus pyogenes, they produce M proteins that prevent phagocytes from adhering and capsule formation. So, polysaccharide capsules in bacteria like Neisseria meningitidis, Yersinia pestis, and Haemophilus influenzae, they hinder phagocyte binding. Also, there is this resistance to digestion. So, blocking lysosomal fusion like Salmonella, Mycobacterium tuberculosis, and leprae, they prevent lysosomes from fusing with the phagosomes. And then another one is the production of toxins. So toxins like leukocidine from Staphylococcus, Listeria O from Listeria monocytogenes, and Streptococcus as streptolysin. These guys, these toxins can cause or can trigger the release of lysosomal contents into the cytoplasm, thereby killing the phagocytic cells. Also, they can evade the complement system by capsule production and complement disruption. So capsules, they prevent the binding of complement component C3b, and C3b is a key component in phagocytosis. Next is complement disruption. So bacteria can produce molecules that interfere with the complement pathways, such as protein H in Streptococcus pyogenes, which binds to C1 and stops the complement cascade. Or there is also this sialic acid in the cell walls of group B streptococci that blocks the complement pathway or inhibits the complement pathway. So let's move to group A streptococci. So characteristics, they're gram-positive, spherical or ovoid or lancet-shaped bacteria, catalyze negative, and often form pairs or chains. And for the classification, classified by cell wall components, primarily M and T proteins into Lancefield groups. We have A through H and K through V, named after Dr. Rebecca Lansfield. And some strains have hyaluronic acid capsules, which enhances resistance to phagocytosis. So the virulence factors of Streptococcus pyogenes or gas or group A Streptococcus. So we have M protein and hyaluronic acid capsule. So for the M protein, that's the main virulence factor composed of two twisted alpha helical chains. Inhibits phagocytosis by its negative charge, and it reduces, how to say that, complement activation by limiting C3 deposition. And the hyaluronic acid capsule, it also contributes to evasion of phagocytosis. And immunity is specific to the M protein serotype. So there's a lot of serotypes existing, over 100. So infection with one serotype does not protect against the other serotypes. We also have exoantigens and exotoxins. So the pyrogenic exotoxins A, B, and C, they are involved in scarlet fever rash, and they contribute to pathogenicity. We have enzymes and toxins like streptolysine O, they affect the erythrocytes, breakage of DNA, DNase B, hyaluronidase, degradation of hyaluronic acid. We also have NADase or nicotinamide adenine dinucleotidase, which is involved in metabolism and conversion of plasminogen to plasmin, which aids in fibrinolysis, that's the streptokinase. For the diagnostic methods of gas or group A streptococci, we have culture and rapid screening. And Commonly, they are utilized for the early detection of infection. While for the antibody detection, this is vital for, it's key for diagnosing complications like glomerulonephritis and acute rheumatic fever. And laboratory methods focus on detecting antibodies to streptococcal products like enzymes and exotoxins. So, clinical manifestations of gas infection, GAS infection, overview of infections. So, Streptococcus pyogenes causes a range of infections from mild to life-threatening. So, the primary infection sites are the upper respiratory tract and the skin, with pharyngitis and impetigo, this one being the most common. So common clinical manifestations, as pharyngitis. In pharyngitis, we can observe this one, pharyngitis, fever, chills, severe sore throat, headache, tonsillar exudates, petechiae on the soft palate. We can observe the petechiae here, and anterior cervical lymph adenopathy. For impetigo or streptococcal pyoderma, this one, a skin infection, of course, particularly in young children, marked by vesicular lesions that turn pustular and crusted. Usually found on extremities, but you can also observe it on the child's face or the patient's face. Other acute or suppurative complications, we have otitis media and ear, scarlet fever, erysipelas, cellulitis, sepsis, sinusitis, acute bacterial endocarditis, and meningitis. So for the severe invasive infections, we can have two examples, TSS and necrotizing fasciitis. So for toxic shock syndrome, this one, of course, is a life-threatening multi-system condition, which it just starts as a skin or soft tissue infection, and then it can lead to shock and renal failure and due to excessive cytokine production. For necrotizing fasciitis, this one is a rapidly spreading skin infection that invades the muscles and deep fascia, causing ischemia, tissue necrosis, and septicemia. And this condition is, of course, a medical emergency requiring very immediate treatment. One damaging sequelae of gas infection is acute rheumatic fever, so ARF. It happens in 2 to 3% of individuals after pharyngitis or tonsillitis, but not in skin infections. And then symptoms include fever, joint pain, and heart inflammation, usually appearing 1 to 3 weeks post a sore throat. And this is likely caused by cross reactivity between antibodies or cell mediated immunity against streptococcal antigens and that of the human heart tissue, particularly the M protein. Another damaging sequelae of gas infection is post-streptococcal glomerulonephritis. So this is a condition characterized by kidney glomeruli damage following skin or pharyngeal infections, and most common in kids aged 2 to 12, especially during the winter, in winter months in the US. Symptoms include hematuria, proteinuria, edema, and hypertension. And this condition is believed to be the result from immune complex deposition. Antibody streptococcal antigen, that's the component of the immune complexes in the glomeruli, triggering an inflammatory response that really can harm the kidneys or damage the kidneys. So for the detection of gas antigens, number one, we have culture-based diagnosis. So the gold standard for diagnosing acute streptococcal infection is to culture the organism from the infected site. and the specimens are plated on blood agar and are incubated. So group A, gas forms small translucent colonies with a clear zone of beta hemolysis. And then identification is typically based on susceptibility to bacitracin, pyre activity or PYR, or growth in the presence of trimethoprim sulfamethoxazole. Another method of detection is rapid antigen detection test or RAD-DS. So commercial kits use throat swabs to detect gas antigens via enzymatic or chemical extraction followed by EA or enzyme immunoassay or latex agglutination. Red teas are quick, needing only like 2 to 5 minutes of hands-on time, but their sensitivity can vary. While they have high specificity, guys, cultures are recommended for negative RAD-T results only. So now we are on the molecular methods for detecting gas antigens. So advanced methods like real-time PCR and hybridization of specific rRNA sequences have been developed to rapidly. So if it's a molecular method, it's always rapid. And these methods offer high sensitivity and can be particularly useful in situations where quick diagnosis is critical. Next is serotyping and molecular typing. So serotyping involves identifying M protein antigens with type-specific anti-sera, but it has limitations, including the availability of typing sera and the emergence of new M types. And for genotyping, such as PCR amplification of the EMM gene followed by sequence analysis, really it can provide a more reliable alternative. We also have FPGE or pulsed field gel electrophoresis. It is utilized in epidemiological studies to create a unique DNA fingerprint for comparison during gas outbreaks. So what's the importance in diagnosis or how important diagnosis is? So serological diagnosis is crucial because the organism may not be present at the infection site by the time symptoms appear, especially in rheumatic fever and glomerulonephritis. And then exotoxins and antibody response. So group A streptococci produce more than 20 exotoxins, and the antibody response to these toxins is used to document non-suppurative diseases. So the key exotoxins, so the notable exotoxins include streptolysins ONS, DNases ABCD, streptokinase, NADase, and others. So the diagnostic antibodies, the most diagnostically important antibodies are of course ASO, anti-DNase B, anti-NADase, anti-hyaluronidase, NADase or NADase. And tests for these antibodies can be done individually or using kits like streptozyme, but streptozyme is a slide agglutination test, which detects antibodies to multiple streptococcal products. So for the serological evidence of disease, elevated or rising titers of streptococcal antibodies indicate disease, with the onset of clinical symptoms typically coinciding with a peak antibody response. So a significant indicator or finding is a fourfold increase in antibody titer acute, between paired acute or convalescent sera. And testing for ASO and anti-DNase B is most common, as not all patients produce detectable ASO levels. And anti-hyaluronidase tests are less common and usually performed in research or reference labs. So ASO testing or anti-streptolysin, O or ASO testing. So that was the purpose and methodology. So ASO tests detect antibodies to streptolysin O, which is an enzyme produced by gas that lyses RBCs. And then the presence of these antibodies indicates a recent streptococcal infection. especially in patients suspected of having acute or rheumatic fever or post-streptococcal glomerulonephritis after a throat infection. So for the classic hemolytic method for the ASO testing, sorry guys for this typo error. So the traditional ASO titer test involves diluting the patient's serum and adding streptolysine or reagent. And after incubation, the RBCs are added as indicators. And if there's enough antibodies are present, streptolysine O is neutralized and no hemolysis happens. And the titer is reported as the highest dilution showing no hemolysis. And then the results may be expressed in TOD units or international units or IU, depending on the standard utilized. So ASO testing based on nephelometric methods. So due to labor-intensive nature of the classic uh, method, modern ASO testing usually employs nephelometry, which is an automated process that quickly provides quantitative measurements of ASO titers. So in this method, purified recombinant streptolysine antigen combines with the antibodies in the patient's serum to form immune complexes, so leading to increased light scatter, which is then converted to peak rate signal. So for the interpretation of the ASO testing results, so normal ASO titer values vary based on factors such as age, geographic location, and season. So moderately elevated titer at least 240 TUD units in adults, and then 320 TUD units in children. So ASO titers typically rise within 1 to 2 weeks after infection and peaking between 3 to 6 weeks after the onset of symptoms. And however, 15% of patients with ARF or acute rheumatic fever may not exhibit an antibody response during this time. So ASO titers, generally they do not increase in cases of skin infections. Next, anti-DNASB testing is particularly valuable in diagnosing post-streptococcal glomerulonephritis after skin infections, as ASO antibodies are often not produced in these cases. And then this test, anti-DNSB testing, is also helpful for patients with acute rheumatic fever who test negative for ASO. And since DNase-B is primarily produced by gas, this test is highly specific for detecting streptococcal sequelae. Again, highly specific for detecting streptococcal sequelae, that's your anti-DNase-B testing. So testing methods for anti-DNase-B testing, so various methods including macrotiter, macro and micro, EA, and nephelometry have been developed for anti-DNase-B testing. So the classic method relies on neutralization, where anti-DNase-B antibodies inhibit reagent DNase-B from depolymerizing DNA. So the presence of DNase is assessed by its impact on DNA methyl green conjugate with the color intensity indicating the results. So interpretation of results for anti-DNase-B testing. So normal anti-DNase-B titers for children 2 to 12 years range from 240 to 640 units. Results from nephelometric or methods are reported in IU or international units per ml. And these are extrapolated from values obtained using the classic method. And another form of testing, we have streptozyme testing. So the streptozyme test, it is a slide agglutination screening test or method that detects antibodies to various streptococcal antigens, including streptolysine, streptokinase, hyaluronidase, DNase, NAdase, and then the sheep RBCs. are coated with these antigens and are mixed with a 1 is to 100 dilution of patient serum. And then hemagglutination indicates a positive result, confirming the presence of antibodies to one or more streptococcal antigens. So for the limitations and usefulness, so although the streptozyme test is quick and easy to perform, it is less reproducible than ASO and anti-DNase B tests, with a higher incidence of false positives and negatives. While it can detect a wider range of antibodies, single titer results are less meaningful than serial titrations conducted weekly or biweekly after symptom onset. And the streptozyme test is a valuable screening tool, but it should be used alongside ASO or anti-DNase B tests when gas sequelae are suspected. So now another microbe or bacterium. We have Helicobacter pylori infection. Characteristics and detection methods. So overview, H. pylori or Helicobacter pylori is a gram-negative microaerophilic spiral bacterium that is the leading cause of gastric and duodenal ulcers. And then it affects 30% of individuals in developed countries and over 90% in developing countries. So untreated H. pylori infections can persist for life and may result in gastric carcinoma or gastric cancer. So its genetic characteristics. So the bacterium exhibits or shows considerable genetic diversity or genetic heterogeneity due to frequent exchange of genetic material between strains. And then two major virulence factors include the highly immunogenic protein CA-CAG-A or CAG-A, CAGA, which disrupts cell signaling and cytoskeletal structures, and the VAC-A gene or VACA gene, which encodes a toxin precursor. So strains containing these genes have a higher risk of causing severe conditions such as gastric ulcers and gastric carcinoma. So for H. pylori, we have invasive testing methods and we also have non-invasive methods. So under invasive testing, we have endoscopy-based methods. So culture. So though highly specific, culture sensitivity is limited as H. pylori may not be evenly distributed in tissue samples. And for the histological examination, gastric biopsy tissues are analyzed to detect the presence of H. pylori. And for the urease biopsy test, and since H. pylori produces urease, this test detects bacterial presence by monitoring pH changes due to urea breakdown, and results can be available within 2 hours. It's fast. Now about let's talk about non-invasive testing, the UBT or the urea breath test. So basically, the patient ingests urea labeled with either radioactive carbon 14 or the non-radioactive carbon 13. So H. pylori metabolizes urea into ammonia and bicarbonate, and bicarbonate is exhaled as labeled CO2. And this test is sensitive, specific, and is helpful in confirming bacterial eradication post treatment or after treatment. But however, the bad thing if it's using the radioactive carbon 14, so that's the disadvantage. So non-invasive testing, we have EA. This is the fecal sample, stool sample, 0.5 grams, and then it will undergo an enzyme immunoassay procedure. So this is for fecal samples, looking for fecal antigens. So stool samples are tested for H. pylori antigens using enzyme-labeled monoclonal antibodies, and this method is mainly used to confirm bacterial eradication after treatment. And its sensitivity and specificity range from 84% to 95% and 97% respectively. So non-invasive testing, molecular tests, we have PCR-based methods to detect H. pylori, so DNA in fecal samples. And however, these tests cannot distinguish between live and dead bacteria. So there's a great possibility that it can result to false positives, especially if in the sample there is the residual DNA after treatment. And then real-time PCR with attachment technology is used, or the thermos aquaticus is used to determine bacterial load and has shown good correlation with the urea breath test. So let's go to your Mycoplasma pneumoniae infections. Mycoplasma pneumoniae, they belonging to the class Molecutis. So it's notable for its lack of cell walls, small genome, presence of sterols in their cell membranes, and complex growth requirements. So these characteristics or features make the culturing of this organism difficult and really time consuming. So for the overview, M. pneumoniae is a leading cause of upper respiratory infections worldwide, and symptoms include sore throat, chills, hoarseness, tracheobronchitis, and headache. So it accounts for 20% of community-acquired pneumonia or CAP and hospitalizations for pneumonia in the US. So culture. So due to the absence of a cell wall, M. pneumoniae is sensitive to drying. So the transport and preservation for the is really crucial, guys. And then, however, culturing is not commonly performed from respiratory samples because it is difficult. So culture is not suggested for Mycoplasma pneumoniae. So serological testing often indicates infection before culture results are available. So let's use serological testing. So here, historically, diagnosis relied on testing for cold agglutinins, which are autoantibodies that cause RBCs to clump or agglutinate at temperatures below 37 degrees Celsius. However, this method is no longer recommended due to its lack of specificity. So modern serological tests include EA, particle agglutination, indirect immunofluorescence, and complement fixation. And these tests can detect IgM, IgA, and IgG, antibodies specific to M. pneumoniae. And EA is the most widely utilized due to its sensitivity and ability to process large volumes of samples. So testing for both IgG and IgM is recommended for improved or greater accuracy. So this one, very pretty, the IFA, indirect fluorescence assay, and it can detect either IgM or IgG antibodies. So in this test, mycoplasma antigens are fixed into a gas slide, and the patient's serum is incubated with these antigens. So after washing, fluorescent labeled anti-IgM or anti-IgG is added, and the slide is observed under a fluorescent microscope. So if the test is positive, serial dilution may be performed to assess antibody concentration. PCR testing is also one of the methodologies, of course, molecular, that can be used to detect mycoplasma. So PCR is a fast and sensitive technique for detecting M. pneumoniae in various samples such as throat swabs, nasopharyngeal swabs, sputum, fixed tissues, and cultures. And it can identify an infection using a single specimen, with just one specimen, no, without needing live organisms. However, the presence or the presence or inhibitors present in nasopharyngeal aspirates may cause false negative results, so requiring additional testing if PCR yields a negative outcome. And for the RNA amplification methods, these methods are highly sensitive because M. pneumoniae cells contain numerous ribosomal RNA copies, indicating active organisms. So the presence of ribosomal RNA copies, meaning these organisms are still alive or active, and these techniques target several genes, including the ATPase operon gene, the PI adhesion gene, the 16S RNA gene, and the TOF gene. And then molecular testing is anticipated to become the preferred diagnostic approach for pneumonia infections. So eventually it might replace serological testing in this case for pneumonia. And now we are down to the last group of bacterium. So Rickettsial infection. So characteristics, the Rickettsiae are short rods or Helicobacilli. They are obligate intracellular gram-negative bacteria. And they are, Rickettsia is divided into two distinct groups. We have the SFG and TG. So spotted fever group and the typhus group. So each responsible for different diseases. So these diseases are often linked to arthropod hosts, including ticks, mites, lice, or fleas, which are essential for the organism's life cycle before transmission to humans. And then generally, humans are accidental hosts, except for Rickettsia prowazekii, the cause of epidemic typhus. So certain species of Rickettsia are geographically restricted, like for R. japonica, only in Japan, and in the Western Hemisphere, we have R. Rickettsii, and global distribution or worldwide, we have R. typhi. So in the United States, the main rickettsial diseases include endemic typhus and the epidemic typhus. Endemic caused by R. typhi and epidemic by R. filis. And we also have the RMSF or the Rocky Mountain Spotted Fever caused by R. rickettsii. So symptoms typically emerge about 7 days after a tick bite and include fever, severe headache, myalgia, nausea and vomiting, abdominal pain, and coughing or cough. And then there is this characteristic rash that develops on the hands and feet, spreading to the trunk after 3 to 5 days of the initial symptoms. So the infection targets endothelial cells, leading to increased vascular permeability and focal hemorrhages or localized hemorrhages. So for disease symptoms for murine typhus caused by R. typhi, so the condition is marked by a cough and chest infiltrate, indicating pneumonia, and a rash is observed in approximately half of the cases. And severe manifestations or symptoms may include seizures, coma, and respiratory failure. So rickettsial diseases are deadly. And then the diagnostic methods. So serodiagnosis is the preferred method for detecting or identifying rickettsial infections, employing techniques such as IFA, micro-IF, IPA or immunoperoxidase assays, ELISA or enzyme-linked immunosorbent assays, and immunoblot assays or IBA. So previously, the Wheel Felix test was utilized to detect antibodies based on cross-reactivity with Proteus species. But this method is now considered outdated due to its lack of specificity. And then now we have IFA and micro-IF. They are considered as the gold standard for detecting rickettsial antibodies. So this is an example of the result. coming from a patient positive for Rickettsial infection. And then IPA is also highly sensitive and specific using light microscope instead of a fluorescent one. But our IFA and micro IF, this is just one of the examples of the results from our IFA and IF. And then these methods can identify significant antibody titers of diseases like RMSF or the Rocky Mountain spotted fever by the second week of infection. However, treatment should begin by the fifth day of illness for effectiveness. So making serology more useful for retrospective diagnosis. So for the molecular techniques, we have PCR. If you wanted to diagnose your Rickettsial infection more rapidly and accurately, and specific genes targeted for PCR include the citrate synthase gene, 17 kilodalton antigen gene, both for SFG and TG, and only for SFG we have the OMPA and OMPB. So PCR can be followed by restriction digest, nucleotide sequence analysis, or sequencing analysis of the amino acids, or real-time PCR for species identification. And the advancement of real-time PCR enhances diagnostic capabilities for rickettsial diseases. So in summary, for group A streptococcus, so this bacterium can be cultured for quick or rapid detection. However, complications like ARF, acute rheumatic fever, and post-streptococcal glomerulonephritis can arise, guys, even after the bacterium is no longer present in the body. And detecting antibodies like anti-streptolysin O and anti-DNase B is essential for identifying these complications. So testing methods include NEF or nephelometry, latex agglutination, and enzyme neutralization. For Helicobacter pylori, so this bacterium leads to gastric ulcers and heightens the risk of gastric carcinoma or cancer if left untreated with antibiotics. So treatment is really essential. Detection methods include or encompass invasive tests such as urease detection from gastric biopsy tissue and non-invasive tests like breath analysis following urea ingestion. So the UBT, urea breath test. And the serological procedures, including ELISA and rapid tests, can identify antibodies, while PCR can detect H. pylori DNA in fecal specimens or fecal samples. And for Mycoplasma, pneumonia, and Rickettsial, so historically, guys, the serological testing has been the standard for these infections, often based on cross-reactivity with non-specific antigens. So in the case of M. pneumoniae, cold agglutinins reacted with the I antigens on RBCs, and for rickettsial infections, the wheel Felix test, which involves agglutination using Proteus bacteria strains, was commonly utilized. However, modern serological methods are more sensitive and specific to the organism, and molecular techniques like PCR are really promising for future diagnostics. So that's the end. Thank you for your attention, and please give your best on your studies. Goodbye.